everyone. Myself Manju Sharma from Suresh Kandiya University, Jagatpura, Jaipur. Department is School of Agriculture. Today we are going to talk about history of plant virology and transmission of plant viruses. History of plant virology. Plant virology 1886 Adolf Meyer which described a disease of tobacco that is called Mosaic Karen had and that is tobacco mosaic. Adolf Meyer demonstrated the safe transmission of tobacco mosaic virus disease. In 1892, Dmitry Lewandowski demonstrated that the causal agent of tobacco mosaic could pass through bacterial filter. 1895, E.F. Smith of USA showed the peach yellows was a contagious disease. In 1898, M.W. Beijering, a Dutch microbiologist and founder of virology, proved that the virus inciting tobacco mosaic is not a microorganism. He believed it to be contagium vivum fluidum, that is, an infectious living fluid. He was the first to use the term virus, which is the Latin word for poison. In 1929, F.O. Holmes provided a tool by which the virus could be measured by showing that the amount of virus present in a plant sample preparation is proportional to the number of local lesions produced and appropriate host plant leaves rubbed with the contaminated sap. In 1935, W.M. Stanley proved that viruses can be made as crystals. He got Nobel Prize in 1946. In 1936, F.C. Bowden and N.W. Pyrie, that is a Britain, found that the crystalline nature of the virus contains nucleic acid and protein. In 1939, Cauchy and colleagues first time showed that the TMV virus particles with the help of electron microscope. In 1956, Gierer and Scarum proved that the nucleic acid fraction of the virus is actually the infectious agent. In 1959, Monday succeeded in including TMV mutations. In 1966, Cassanis, which discovered the satellite viruses. In 1971, T.O. Diner discovered viroids which only consist of nucleic acid, smaller than viruses, caused potato spindle tuber disease, 250 to 400 bases, long of single standard circular molecule of infectious RNA. After that, virus. Viruses are ultramicroscopic nucleoprotein entities which are infectious agent and obligatory parasitic pathogen which are less than 200 μm in size. They are devoid of enzyme and depend on the host protein synthesis machinery, that is ribosome. So they have only one type of nucleic acid via RNA or DNA. Most of the plant virus is having RNA, for example TMV. Few viruses contain DNA, example cauliflower mosaic virus, which contain the DNA as a genetic material. Virus, banana bunchy top virus, maize streak virus, and sugar beet curly top virus. Viroids. Viroids are a small, low molecular weight ribonucleic acid that can infect plant cells, replicate themselves, and cause the disease. So they are also called as mini viruses. Example: potato spindle tuber, chrysanthemum, stun, and coconut cadding. Without a host cell, viruses cannot carry out their life-sustaining functions or reproduce. They cannot synthesize proteins because they lack of ribosome and must use the ribosome of their host cells to translate viral messenger RNA into viral proteins. Viruses cannot generate a store energy in the form of adenosine triphosphate. Viruses cannot generate or store energy in the form of adenosine triphosphate ATP, but have to derive their energy and all other metabolic functions from the host cell. They also parasitize the cell for basic building materials such as amino acids, nucleotides, and lipid fats. Although viruses have been speculated, speculated as being a form of proto-life, their inability to survive without living organisms makes it highly unlikely that they proceed cellular life during the Earth's early evolution. Some scientists which speculate that viruses started as rogue segments of genetic code that adapted to a parasitic existence. All viruses contain nucleic acid, either DNA or RNA, but not both. Some scientists which speculate that viruses started as rogue segment of genetic code that adapted to a parasitic existence. Then all viruses contain nucleic acid, either DNA or RNA, but not both. and a protein caught, which in case is the nucleic acid. Some viruses are also enclosed by an envelope. Some viruses are also enclosed by an envelope of fat and protein molecules in its infective form. Outside the cell, a virus particle. Some viruses are also enclosed by an envelope of fat and protein molecule in its infective form. Outside the cell, a virus particle is called a virion. Each virion, which contains at least one unique protein synthesized, each virion contains at least one unique protein synthesized by specific genes in its nucleic acid, meaning then, in its infective form outside the cell, a virus particle is called as virion. Each virion contains at least one unique protein synthesized by specific genes in its nucleic acid, like meaning that is virus-like are disease-causing organisms that contain only nucleic acid to have. Each virion contains at least one unique protein synthesized by specific genes in its nucleic acid, that is, the virus like and disease causing organism. That too only. Uh, then the other virus like particles called prions are composed primarily of a pathogen protein tightly integrated with a small nucleic acid molecule. Then the viruses are generally classified by the organism they infect, animals, plants, or bacteria. Then animals, plants, or bacteria, since viruses cannot penetrate plant and cell wall. Then virtually all plant viruses are transmitted by insect or other organisms that feed on plants. Then the nature of viruses was not understood until the 20th century, but their effects had been observed from 4 centuries. British physician, that is Edward Jenner even. So the British physician, that is Edward Jenner, even discovered the, the principle of inoculation in the late 18th culture. So after he observed that people who contracting the mild cowpox disease were generally immune to the dead layer smallpox disease. By the late 19th century, scientists knew that some agent was causing a disease of tobacco plants but would not grow on an initial growth on an artificial medium like bacteria and was too small to be seen through a light microscope. Then, the advances in live cell culture and microscopy in the 20th century eventually allowed scientists. Then, advances in live cell culture and microscopy in the 20th century. So, by the late 19th century, the scientists knew that some agent was causing a disease of tobacco but would not grow on an artificial medium. So, the advances in live cell culture and microscopy in the 20th century eventually allowed scientists to identify virus. Then, advances in genetics dramatically improved the then advances in live cell culture and microscopy in the then the advances in genetics dramatically improved that identification process is complete. So the advances in genetic dramatically by the late 19th century, the scientists knew that the some great was causing a disease of tobacco plant. So by the late 19th century, scientists knew that some agent was causing a disease of tobacco plant but would not be grown on an artificial medium. By the late 19th century, scientists that knew some agent was causing a disease of tobacco plant but would not grow an artificial medium. So, by the late 19th century, the scientists knew that the some agent was causing a disease of tobacco plants. Someone where then the advances in lifestyle culture and microscopy in the 20th century eventually allowed to then advances in genetic dramatically improved the identification process. So that is the figure of animal viruses. So here the capsid will be found, DNA, and the spikes are also found. Then capsid. The capsid is the protein shell that encodes the nucleic acid, but uh, the capsid is the protein shell that encloses the nucleic acid. With its enclosed nucleic acid, which is called the economic importance, and with this enclosed the nucleic acid, it is called the nucleocapsid. Then the envelope. Then the envelope. Many types of virus have a glycoprotein envelope for many types of virus have a glycoprotein envelope surrounding the nucleocapsid. The envelope is recounted as many viruses have a glycoprotein envelope surrounding of nucleocapsid. The envelope is compared with two The envelope is composed of two lipids layer interpressed with protein molecule, that is lipoprotein bilayer, and may contain material from the membrane of the host cell as well as host viral origin. The nucleic acid. So the envelope is composed of two lipid layers interpressed with protein molecules, that is lipoprotein bilayer, and may contain material from the membrane of the host cell. Then the nucleic acid, just as in cells, the nucleic acid of each virus encode the genetic information for the synthesis of all proteins, while the double-stranded DNA is responsible for this in prokaryotic and eukaryotic cells. Only a few groups of USA virus use in DNA. Nucleic acid, just as in cells, the nucleic acid of each virus encodes the genetic information for the synthesis of all proteins, while the double stranded DNA is responsible in this in prokaryotic and eukaryotic cells. Only a few groups of viruses use DNA envelope. Many types of virus have a glycoprotein envelope surrounding the nucleocapsid. The envelope is composed of two lipid layers interpressed with protein molecules like lipoprotein bilayer and may contain material from the membrane of a host cell as well as that of viral origin. Capsid. The capsid is the protein cell that encloses the nucleic acid. With its enclosed nucleic acid, it is called the nucleocapsid. Parasites that do not have the molecular machinery to replicate without a host. Plant viruses can be pathogenic to higher plants. Most plant viruses are rod shaped with protein discs forming a tube surrounding the viral genome. Isometric particles are another common structure. They rarely have an envelope. Then the tobacco mosaic virus or TMV. TMV is a positive sense single stranded RNA virus in the genus that is Tobamovirus that infects a wide range of plants, especially tobacco and other members of the family Solanaceae. The infection causes characteristic patterns such as mosaic like mottling and discoloration on the leaves, hence the name. TMP was the first virus to be discovered, although it was known from the late 19th century that a non-bacterial infectious disease was damaging tobacco crop. Then transmission of viruses. Firstly, the insect vectors which feed on infected plants and acquire virus. Then the virus persists in the vector and travel with the vector to a new plant host. Then the third thing, virus inoculation occurs when the insects feed on the plant and deliver virus into the plant cell. Then the mechanical transmission of viruses through the sap. Here, the transmission of a virus from infected to healthy tissues is a procedure fundamental to the study of virus diseases. In the laboratory, this is usually accomplished by grinding the leaf of a diseased plant and rubbing the infectious sap on the leaf of a healthy plant. After a strong wind injures the leaves of adjacent disease and healthy plants, after a strong wind injures the leaves of adjacent diseased and healthy plants, or when plants are wounded during cultural practices by tools, hands, or clothes, or by animals feeding on the plant, and the sap carrying virus is transferred to wounded plants. So here, disease through the natural leaf contact and rubbing. Then one plant which is the diseased plant, and the another plant which is healthy plant. So through the natural leaf contact and rubbing the plant leaf, there will be the transmission of the virus. So one plant is the diseased plant and the another is the healthy plant. So throughout the handling, the healthy plant which will be also infected by the same disease or this kind, there is the transmission of the same virus into the plants, between the plants. Then there is the plant virus life cycle. First thing, like host cell is there on which the virus are attached. After attachment, the virus which have DNA or RNA genetic material which will be incorporated into the host DNA. Then, in the host, there will be the replication of the viral genome, and after that, after replication, there will be assembly of the genome, and then the particular genome which will be released. And after that, there will be transmission, and this kind from one infected plant the all the plants which will be infected, and this kind there will be transmission of the same kind of virus. Plant viruses are viruses that affect plants. Like all other viruses, plant viruses are obligate intracellular parasites that do not have the molecular machinery to replicate without a host. Plant viruses can be pathogenic to the higher plant. Most plant viruses are rod-shaped with protein disc forming a tube surrounding the viral genome. So there is isometric particles are another common structure. They rarely have an envelope. The great majority have an RNA genome which is usually small and single-stranded. Which is usually small and single-stranded, but some viruses have double-stranded RNA, single-stranded DNA, or double-stranded DNA genomes. Although plant viruses are not as well understood as their animal counterparts, one plant virus has become iconic, that is tobacco mosaic virus TMV, the first virus to be discovered. So basically the plant viruses are the viruses that affect the plants Like all other viruses, plant viruses are obligate intracellular parasite. Obligate, that means the viruses which always require a living host for their survival. If there is no availability of the living host, in that case the viruses which can't grow and they can't live. So they do not have the molecular machinery to replicate without a host, because the viruses which always use the host machinery, and they always use the host body for their replication. So most plant viruses are rod shaped with protein disc forming a tube surrounding the viral genome. Isometric particles are another common structure, and they rarely have an envelope. So basically, there is the great majority have an RNA genome, which is usually small and single stranded, but some viruses have double stranded RNA, single stranded DNA, or double stranded DNA genomes. Although the plant viruses are not as well as understood as their animal counterparts, one plant virus has become iconic. That is tobacco mosaic virus TMV, the first plant virus which was to be discovered. So that is the structure of the like animal virus structure is there, in which the capsid is there, spikes are there, and the DNA genetic material. So through the spikes, basically they will be attached with the host. Capsid is the protein shell that encloses the nucleic acid. With its enclosed nucleic acid, it is called the nucleocapsid envelope. Many types of virus have a glycoprotein envelope surrounding the nucleocapsid. The envelope is composed of two lipid layers interspersed with protein molecules like lipoprotein bilayer, and may contain material from the membrane of a host cell as well as that of viral origin. Nucleic acid, just as in cells, the nucleic acid of each virus encodes the genetic information for the synthesis of all proteins, while the double stranded DNA is responsible for this in prokaryotic and eukaryotic cells. Only a few groups of viruses use the DNA. Otherwise, mostly the viruses, those have RNA.